Vicky, and also Kate for the invitation to discuss with you our recently published data. Can you hear me?
Yep.
Okay, I share the screen. And I'm also happy that actually it is the International Women's Day here. It is evening, it's still. I'm happy that we're going to discuss about science on this special day. So I think this is the best way to acknowledge the contribution also of women to the progress of knowledge. So, do you see the screen?
Yep.
Okay, so I'm going to talk about artificial intelligence applied to blood transcriptomics in multiple sclerosis. This is a project which has been developed by Massimo, who is also connected with us this evening. So, and before going directly into that, we had for this study three main premises. First of all, we know that the immunopathogenesis of MS does not rely on a single cell type. So we do need interaction between multiple cell types. Will you see the arrow while I'm moving it?
Yep, we can see it. Yeah.
Okay, so we need the interaction between innate and adaptive immune system, and then the involvement of the cells which constitute the blood-brain barrier and the CNS parenchyma. There is a complexity of events which have been partly identified in the animal models. So we have some information also regarding the activation steps and phenotypes that cells need in order to establish a neuroinflammatory reaction in the CNS. Then, although out of fashion, we do know that multiple sclerosis does have distinct clinical curves, and the degree of misdiagnosis is quite high, especially for primary progressive MS, whose MRI findings overlap with relapsing remitting MS or other disorders. Further, MS prognosis is poorly predictable, so let's think of the transition from relapsing remitting to secondary progressive MS. So artificial intelligence so far has been mainly applied to MRI data in multiple sclerosis and has shown to be effective in distinguishing MS subjects from healthy donors. That is not a difficult task. Clearly, you have plaques on one side and no plaques on the other side. But when the same tools have been applied in order to stratify distinct clinical courses, they showed limited power. So there are no main MRI features distinguishing relapsing remitting from progressive cases. The third consideration regards therapeutics. So we know that the currently approved drugs target peripheral immunity. So this is the formal proof that peripheral immunity is sustaining disease expression and maintenance. On the other hand, most of them actually are effective in relapsing remitting MS and not in progressive MS. And so probably this is also telling us that peripheral immunity in progressive MS may be different. So we started performing blood transcriptomics in MS many years ago. We started with the relapsing remitting population, for which we provided a gender-based PBNC transcriptome description initially, and then we described also specifically the transcriptomic changes occurring in B cells and monocytes. Meanwhile, we started collecting also samples from clinical isolated syndromes, primary and secondary progressive MS. And in the last years, we published a couple of studies where highlighting, for example, this regulation of interferon signaling in human and experimental MS, or this regulation of MS risk genes. So if we take this last study as an example, here, what we did was to compare PBNC transcriptomes to the transcriptome of healthy subjects. And so we had the CIS, relapsing remitting, primary progressive, and secondary progressive transcriptomes. And you can see there is a subset of MS risk genes which are up in red or down in green regulated under disease compared to the healthy population. But actually, several other MS risk genes were dysregulated in 3, 2, or single stages of or forms of MS. So this is telling us that the alteration in the expression of MS risk genes is not a stable feature reproduced throughout the distinct phases and forms of disease, but may be a selective event for each disease stage. So can we then use the information to predict MS forms from blood transcriptome? And to go into that, we collected many more gene expression profiles relative to the PBNC from more than 300 subjects. And we divided these profiles in a training set used for algorithm benchmarking and optimization. And after that, the training set was useful to build predictive models. And these models were then tested on a completely independent cohort of subjects. Now, to do that, we had to pull data, transcriptomic data generated with different microarrays. And so there has been a lot of work with the pre-processing steps to go into that and have an integrated data set. And so we extrapolated about 10,000 probes, so 10,000 transcripts, relative to more than 300 samples with 57 CIS, 108 relapse remitting, 35 primary progressive, 26 secondary progressive, 60 healthy controls, and 27 subjects with other neurological disorders. I need to mention that all the MS subjects were devoid of treatment, including the progressive cases. Just a few of them had received some immunosuppressive treatment, but many years before. So we then checked whether so-called pre-processing was correct, because along the years we loaded the same RNAs on multiple arrays a long time. And so while before batch correction, only few technical replicates paired together, after batch correction, you can see we had the optimal pairing of all the almost all the technical replicates. So the training and the test data sets were coherent one with the other in terms of gender and age distribution, but clearly there were differences between one group and the other. For example, the progressive cases were clearly older, had higher EDSS and longer disease duration. So when looking at the global transcriptome, we didn't find any effect of age and gender. So we couldn't stratify the subjects which were older than 40 differently from those which were younger than 40. And we didn't have a different separation for women and men. On the other hand, when looking at the condition, we had here on the one side the CIS and relapse remitting MS subjects, here the primary and secondary progressive subjects, and on the other hand, here on the other side, the healthy controls and the other neurological disorders, and we had a similar coherent pattern also on the independent test set. Then Massimo designed an informatic platform using iWiMS, which was integrated with the algorithm, some of the algorithms described in Wicca. And we decided to use decision tree-based algorithms, which have the internal feature selection as part of the learning process. And so we used the largely employed random forest, functional tree, and then we applied a technique which is called adaptive boosting to functional tree, because this has been shown to be extremely effective in improving classification in unbalanced data sets. Massimo also built an unbiased framework for the comparison of the distinct machine learning algorithms based on nested cross-validation using exclusively the training cohort. And after that, he constructed a final model which was tested on the independent test set. So which were the questions we had in mind? First of all, we wanted to see whether PBNC transcriptomes were sufficient information to classify MS from non-MS subjects. So we had relapse remitting primary and secondary progressive MS subjects on one side, and healthy and subjects or people with other neurological disorders on the other side. The second classification task regarding whether there was a specific a trait for the very early phase of disease. And so we compare the CIS against all the other groups. And then the third classification task regarding the distinction between progressive and relapsing remitting MS. So I show you here only the final results on the independent test set. All the rest is in the paper. You can see in the paper. So the first question, can we discriminate the MS state from the controls? And so here you can see precision recall curves using the 3 algorithms, and you can see that the area under the curve is extremely good with higher than 0.9, and the blue line is the one relative to the AdaBoost FT algorithm. And when using this algorithm, we could build a predictive model based on 139 probes, which was then applied to an independent test set and classified 50 out of 53 MS subjects correctly, and 21 out of 27 non-MS subjects. So there was, the signature had a very high sensitivity and an intermediate specificity with an overall accuracy, which was close to 90%. Biological teams present in this signature related to chromatin remodeling and apoptosis, which are 2 mechanisms which have been shown to be involved in MS by our group, but also by many other groups. So there was an issue regarding this comparison, and actually regarding all the comparisons we did, because the MS group was a bit older than the non-MS group. And this was a significant difference while the gender distribution was similar. So what we did was to check whether the classification signature was influenced by demographic factors. And as you can see here in these PCA plots, this signature did not distinguish females from males and did not distinguish subjects based on age, but it was very effective in distinguishing subjects based on condition, so MS versus non-MS. Regarding the second task, the identification of early signs of disease, again, here we had the AdaBoost algorithm performing better than the other, but you can see that the area under the curve is only 0.49. So clearly here the algorithms had troubles in finding the right signature to classify the subjects. So this is the first information. There is not a strong signature for the CIS population. And so using that algorithm, again, we had a predictive model based on 213 probes, which classified only 6 out of 9 CIS subjects, but it was quite good in terms of specificity because it classified 74 out of 80 control subjects. And here, again, we do have biological terms related to immune response, interferon signaling, chemotaxis, so all processes that we know to be involved in MS. This signature did not distinguish subjects based on gender and age. And you can see that it distinguished subjects based on condition, but only not according to the first component, but only when we added the second component. Then regarding the third task, the identification of progressive MS from relapsing remitting MS. First of all, we pooled primary and secondary progressive MS together and compared them to relapsing remitting MS. You can see here again, the areas under the curves are extremely good. And we did find the same with the AdaBoost FT algorithm, also when we looked at primary progressive or secondary progressive alone versus relapsing remitting MS. So when we applied the AdaBoost algorithm, we looked at the combined progressive population, we had a model based again on about 200 probes, which identified 15 out of 18 subjects, progressive subjects, and 34 out of 35 relapsing remitting subjects. So quite good sensitivity and excellent specificity. And again, the classification was very good, although we had a lower number of subjects in the primary progressive compared to the relapsing-remitting group. And so we had 9 out of 10 primary progressive subjects correctly identified and only 1 relapsing-remitting subject misclassified, while the secondary progressive signature had only 1 misclassification in the primary progressive population, but all the relapsing-remitting MS subjects were correctly identified. Now, there is debate in the literature about whether primary or secondary progressive MS are the same or are different states of disease. So if they are the same, we would expect that the algorithm is picking up the same or similar features when comparing each phase to the relapsing-remitting population. But when comparing the contents of the predictive signatures, which are described in the paper, you can see that the overlap between the primary progressive signature and the secondary progressive signature is very low. So clearly the algorithm prefers to choose different features to make the prediction of primary and secondary MS, primary and secondary multiple sclerosis. And so this is also suggesting that indeed there are differences between the 2 states of disease. We also checked whether also the progressive signatures were influenced by demographic factors. They were not by gender. We know that the relapsing-remitting population is younger, significantly younger than the secondary progressive population. But when we use the signature to stratify all subjects included in the study, so also CIS subjects, ONDs, primary progressive, and then the signature was not distinguishing subjects based on age anymore, while the distinction based on conditions, so progressive stage versus relapsing remitting, was again very clear. So in this study, we could show that dissection of MS through blood transcriptomics uncovered distinct changes in MS subjects depending on disease stage, that now these transcriptional alterations may help tracing changes in specific immune cell subsets under disease. And we have also demonstrated for the first time that machine learning approaches for biomarker selection allow the identification of transcriptional signatures predictive of MS state and progression. And these classifiers were largely independent of age and gender. Now, this study can clearly be extended to larger and diverse cohorts. What we are also doing is to try to use different types of algorithms, different types of omics data, and other inquiries in MS and other neurological disorders. For example, last year, we also supported collaborators at the University of Lugano who developed a machine learning model using flow cytometry data relative to extracellular vesicles circulating in plasma. And this information was sufficient to identify Parkinson's disease from healthy subjects. So I want to thank, first of all, Massimo, who developed this study, and previous collaborators, and as Ramesh, Marco, and Sundar. All the work was done at the Institute of Experimental Neurology at San Raffaele Institute in Milan. And this institute has been led by Giancarlo Comi for many years, and I'm very grateful to him. And this work would not be possible without a tight collaboration with the neurology department, which is now led by Massimo Filippi. And I want to acknowledge many other collaborators within my institute and around the world, all of you for the attention, and I was also to wish you a happy International Women's Day.
Thank you very much, Cinthia, for a lovely talk. With your other neurological disorders, were they one specific disease, or were they a mix of
which had different things. So they had, for example, stroke. A few of them had stroke. Others had Parkinson or paraparesis. One had autoimmune encephalitis, migraine. So clearly it is not an homogeneous group of subjects.
Yeah, okay. Okay. So do you think it would be as accurate if you were looking at just a group of patients who had, say, like one of the exclusion diagnoses, like MOG antibody positive, or?
Yeah, that would be absolutely interesting. Absolutely. So we didn't have the chance to collect many samples about that. So that was the reason. So the OND, the mixed OND group was the very first step to go into that.
Okay, thanks. Does anyone else have any questions?
Sorry, I came in a bit late. So what, you use the transcriptomics on what cell types? I missed that bit.
So these were PBMCs.
PBMC, all sorts. I'm still struggling to understand why you couldn't find any difference between for age. I mean, we know that the primary progressive have a later onset. We know that they are 50-50 male, female. So that gender and age didn't separate at all. I just can't get my head around it.
No, the point is that gender and age does not, what we showed at the beginning is that if you look at the whole transcriptome, there is not a strong effect of gender and age on the transcriptome, on the overall transcriptome. And there was also not this effect on the specific signature that the algorithm picked up out of the whole transcriptome. So this was a control that we performed for the signature. This does not mean that there are no differences between men and women with MS. In fact, in our older paper, in the relapsing remitting population, where we compare relapsing remitting to the healthy subjects, but we separated then the women from men. So we compare the healthy women to MS women and healthy men to MS men. We ended up with two distinct signatures, completely different signature, although the clinical parameters for the relapsing remitting form of disease were very similar in the two groups. So the biology behind the clinic may be different in men and women, but you need to look for that specifically if you want to catch it up. Here, the algorithm, at the end, the groups were rather balanced in terms of gender. And so the algorithm was not catching differences based on gender. There were differences based on age, but the predictive signature were not influenced by age. So this does not mean that there are not then other signatures, transcriptional signatures, which trace aging or which trace gender. But this was simply not the right approach to catch them. We had few primary and secondary progressive cases, so we couldn't make a specific analysis for women and for men.
Any other questions? I see, Vilia, you've unmuted yourself.
I have. Hi, Cinthia. Thanks for that beautiful talk.
Hi, Elijah.
Hi. So I'm very interested about the differences between the primary progressive and the secondary progressive cases that you found. And I guess my question really is, can you give us a bit of an insight as to what the differences were between those probes and what that might tell us about the immunobiology of the two different courses?
So all the probes are described in the paper. You find them and you can find also the biological processes. So the main process appearing in secondary progressive MS, for example, regarding blood coagulation. So we search for papers in the literature whether there are described differences between progressive MS subjects and relapsing remitting MS subjects about blood coagulation factors, but we didn't find any. There is one paper from Sven Moit in Germany indicating that indeed there may be specific coagulation factors altered in secondary progressive MS, but not in primary progressive MS patients in comparison with the healthy population. Regarding the primary progressive signature, there we had as main features the ubiquitination processes. So we know that they are related to antigen presentation, and it's clear how relevant is antigen presentation in MS. But the point is that apart from the bioinformatical information that you get, there is not so much investigation about these pathways at distinct stages of disease. So that's why we wanted to describe not only the probes, but also the biological processes, because they do deserve more attention in the comparison among disease stages and forms.
So just to be a bit controversial then, do you think that these are, I guess, pathways and processes that change over time to distinguish between the two courses? Or do you think that there's a completely different underlying pathophysiology between primary progressive and relapse onset MS?
Okay, so already in the paper where we looked at the MS risk genes, we do have indeed distinctive pathways among disease stages. On the other hand, we started also going into the validation of the transcriptional evidences, looking to verify whether some of these evidences actually trace changes in the frequency of specific immune cell subsets. And so, for example, last year, no, in 2019 was that we published a paper where we started from the downregulation of a gene, which is called CD161. We realized that together with this gene, there was downregulation in primary progressive MS of many other genes, which are associated with a specific CD8 positive T cell population, which expresses CD161 at high level. And when we checked the protein levels in completely new cohorts of primary progressive and healthy subjects, we did observe the loss of this population in circulation in primary progressive MS. The same population had been shown already by other groups to be stable or upregulated in relapsing remitting MS. So there may be specific changes in the frequency of certain minor immune cell subsets that we can indirectly catch via the transcriptomic analysis.
Thank you.
Was there any other question? Oh yeah, Helen?
Yeah, no, that was super interesting. Thank you very much. I'm pulling down your paper now, which is probably my answers to my questions are probably in it. But I'm just curious. So, how are you defining your cases? And I suppose how did you enroll your cases and controls, and how did you define MS and CIS? Because I understand that, you know, how you define CIS or capture CIS can sort of differ over time, depending on the diagnostic criteria that's been used.
Yeah, in this paper we had we used the McDonald 2010 criteria for the definition of MS. We collected these samples along many years. And it was not easy, especially for the progressive forms. So now what we are doing is again to further sample primary progressive cases before they get the final diagnosis. And but for secondary progressive subjects, it has become more difficult because in any case where they are undergoing treatment. And so clearly the applied treatments will also induce a transcriptomic dysregulation, and so this could completely then cover the biology behind the different forms of disease. And the clinical isolated syndromes had a very first attack, and we sampled them 1 month after the first clinical attack because a few of them had received steroids. And so we waited 1 month after that. And we sampled them by the 3rd month from the first clinical symptoms. So they were indeed at the very beginning.
And do you happen to know how many of your CIS patients have gone on to transition to fulfilling criteria for MS?
Yeah, clearly we do know for them, but there were too few then to make a distinction between those who convert and those who do not convert. And so for that purpose, we ran another study on a larger population of CIS subjects in order to have the right numbers to distinguish them and to apply machine learning approaches to the transcriptomic profiles.
And sorry, where did your controls come from, or how did you recruit those?
The healthy controls, well, people
Yeah.
around, usually people around in our hospital. They were devoid of any autoimmune and infectious disorders. They didn't take medicaments, and they were matched for age and sex. Well, age mainly for CIS and RR subjects. We had also a few older subjects, but there were many, so around 50, 60 years old, but most of them were anyway around 35.
So was it staff at the hospital with the controls, like lab members and whatnot?
Yeah.
Do you think the differences then between your groups, I don't know, race, ethnicity, socioeconomic status that would be of value to capture in the future, or is that not relevant when it comes to transcriptome? I don't know.
Well, clearly, genetic background and environment change depending on where we live and on our also ethnic origin. So, and so the transcriptome is the result of this interaction. I wouldn't be surprised to find specific predictive signatures, for example, in the African American populations. That should be checked. There is not such a study. We had a quite homogeneous Caucasian Mediterranean population. We didn't use a few samples from Sardinia. You know that Sardinia has a very high incidence of multiple sclerosis, but they do have a very different genetic background compared to mainland Italians. So we prefer not to pull all together. And but it should be tested. Actually, it would be very interesting to try to apply these concepts to distinct ethnic groups.
Yeah, and sorry, apologies taking up time here, but so I'm really interested in the prodromal phase of MS, so the years leading up to MS symptom onset. So I'm just curious, you've obviously got a lot of experience with this platform that you're using. Do you think it has potential value in the future of identifying people in this prodromal phase of disease? And I can see you've been working on Parkinson's, so you're probably acutely aware of this issue. What are your thoughts on that?
So I can only tell you, I think that it will be useful. I can only tell you that we are starting performing preliminary experiments in radiologically isolated syndromes. So also there, it seems there are some traits. The difficulty is to have sufficient numbers then to run machine learning statistics.
Well, it's super exciting. I look forward to seeing your subsequent studies. Thank you very much.
Oh, I thank you for the questions and the interest.
All right, was there any further questions?
I'll ask a question if that's all right.
Yeah, go ahead.
Hi, Cynthia. Great talk. Thank you.
Hi, Kia.
I was just wondering, do you know whether there was any differences in the PBMC makeup between the different groups? Like, are the transcriptomic changes driven by differences in cell function, or the actual differences in the makeup of the PBMCs?
So if you look at the main frequencies of CD4 and CD8 cells, B cells, so the main subsets, monocytes, there are no main differences among the distinct stages of disease. You really need to pick up specific subgroups of cells in order to distinguish them. So no great differences.
Thank you.
You're welcome.
Was there any further questions? Doesn't look like anyone's unmuted themselves. So thank you again, Cinthia, for a wonderful talk. It's really interesting work. Once again, happy International Women's Day to everybody. That's still on Monday.
Yeah, the same to all of you. Thank you very much for the invitation.
Thank you.
Thanks very much.
Thanks,
Bye.
everyone.
Bye. Bye, everyone.
Bye.